All right, so far we've been talking about protein synthesis, okay? We can also call that gene expression, or as we look at this slide, also called the central dogma, okay? We're looking at the flow of information in biology. So we start with our DNA. Once again, that's our blueprint within our cells. It gives us the information that we need to make all of the different types of proteins in our body, but DNA doesn't actually make anything. So when we think about our DNA sequence, once again, we have a series of nitrogen or nucleotides, I should say, that code for particular traits. Okay, and what happens is that we want to protect this DNA. We want it to stay within the nucleus, and we don't want it to get damaged. So we want to switch to another molecule called RNA. And we want a copy of it, and that process is called transcription. So we can take this original message and we can convert it into this copy. Our copy machine is going to convert and pair up adenine with, instead of thymine, uracil. And then we would go and simply complementary base, AUA, okay? And this would be my mRNA strand, and that happened through transcription. That also takes place in the nucleus. These are all the steps that we've talked about for the last couple days. We now take that mRNA message, and that's going to leave the nucleus, the safety of the nucleus, and we can make many, many copies of this mRNA, which are eventually going to go to different ribosomes to actually produce a protein. So we take our sequence, and we actually don't read every single letter, but if you remember, we read in triplets, or these are called codons. All right, and then we can use that handy dandy chart to figure out what different amino acids, and I don't actually have my chart with me right now, are actually going to be linked together to form this protein. Now this is a protein. This gene up here is responsible for flower color, okay? And this particular flower color is going to be pink. And there could be a different sequence, another sequence of nucleotides that would equal a red flower. This is what we've been talking about, the flow of information in biology, that what we are ultimately doing is making proteins, because it is the result of proteins and protein action that we see when we look at all living things. So what we're going to talk about today are mutations. And mutations are simply changes in that blueprint or that genetic message that are going to have a varying effect, or going to have varying effects on an organism. And we're going to see what those effects can be. All right, so a mutation is simply a change in the genetic material. So what we're talking about is at the DNA level. So if we look at all these nucleotides in my little picture here, a mutation would be that if we look at these 3 Gs, they would potentially be deleted. And so everything around it would be red, but that's ultimately going to change, once again, the message in RNA, and then ultimately it's going to change the protein that we produce. So what happens if one of those bases is wrong? Well, it varies. It depends on the type of mutation, and it depends on where that mutation is. So we're going to look at that. All right, so when we think about mutations, changes in genetic material, when we were in our genetics unit, we talked about chromosomal mutations. And these are the result of meiosis. Remember, the making of our gametes, so either egg or sperm. And in meiosis 1, our homologous chromosomes separate, and then in meiosis 2, our sister chromatids separate. And if something goes wrong in that chromosomal separation, then we could either get too many, okay, chromosomes, so plus 47, or we can get too few. So for humans, those numbers would be either plus 47 or plus 45. So with chromosomal mutations, that results in extra or missing whole or parts of chromosomes. So the result are disorders like trisomy 21, so Down syndrome. It could be Turner syndrome, or those other chromosomal disorders that we talked about. What we're going to talk about today are simply gene mutations. And when we think of a gene, we want to think of the DNA. Okay, we want to think of those nucleotides. So it's some sort of change in the nucleotides, either extra letters, okay, or extra nucleotides, missing nucleotides. That the resulting the result is the result is the formation of different alleles. All right, and the different alleles means that a different protein is going to be produced, or that protein is not going to be shaped properly. And remember, we keep talking about this idea of form equals function. If it's not shaped right, it's not going to be able to do its job. All right, the first kind of mutation that we're going to talk about are point mutations. And it's exactly kind of what it sounds like. It's simply a change in one or a few nucleotides. So in your comprehension constructor, this is going to be your first major category, okay? And an example of a point mutation is called a substitution. Now you're going to be asked to make a DNA sequence in the box next to that. So you can make up any sequence you want, or you can use this sequence. But make sure that you understand how to do this and what the result is. So my DNA sequence is simply going to be cytosine, thymine, guanine, adenine, guanine, thymine. Okay? And a substitution is just like what it is in basketball or soccer, is that we're going to substitute, okay, number 19 for number 9. And in our case, we're going to substitute adenine for cytosine. So everything else stays the same. Okay, if you notice here, that's the same right here, and it's the same afterwards. So we just have a single substitution here. Now, what's the effect on this organism? Well, let's take a look at our reading frame over here. So remember, we don't look at every single letter. We read in triplets or codons. We read that genetic message in triplets. So if I have the reading frame, the fat cat ate the rat, and I do a single point substitution. So you look, I substituted my C for an H. All right, my reading frame now reads, the fat hat ate the rat. Did that change the meaning of my sentence? Definitely. We have a hat now eating the rat instead of a cat. It can have varying effects on the organism. The next type of mutation that we're going to look at is called a frame shift mutation, and we're going to go through two separate examples with this one. Frame shift mutations oftentimes have a larger effect on the organism because it shifts the entire reading frame of our genetic message, and we're going to be able to see that here quite easily. The first frame shift mutation we're going to go through is called an insertion. And so once again, you can see my sequence, cytosine, thymine, guanine, adenine. Write an example in your comprehension constructor as well. Guanine, thymine. And I'm going to insert. I'm going to put an extra nucleotide into my DNA sequence. So here was the same, right? Same. I added one in here, and then it's the same afterwards. So how does this affect the organism? Let's go back to our reading frame over here. So the fat cat ate the rat. That's our initial reading frame. And I'm going to insert an extra nucleotide, and I did that right here, okay, right after that arrow there. So now let's see what our new reading frame is. The fat hacha tat eth era t. Did that change our message or our reading frame or our final message? Yes, substantially, okay, to the point that we don't even understand anything after the fat. Okay, so you can see the effects of these frame shift mutations. It affects everything else after that simple insertion, because now we're not getting the same amino acids after this point. Now deletion has the same kind of effect, all right, except it's exactly what it says. Instead of adding, we're going to delete one. So here's our initial. Let's see here, where do we delete one? Oh yeah, here's our initial, and we're going to delete this adenine here. Okay, so here's our initial nucleotide sequence. We delete that adenine, and it shifts our reading frame to the left. So once again, our original kind of sentence is the fat cat ate the rat. With that deletion, we had the deletion happen right here, removing that H. And now what we have is tef atka ada tet gerat. I have a hard time even saying that. So now we can't even make any sense of what would be a protein, or in our situation, this sentence. So once again, frame shift mutations oftentimes have a more dramatic effect on that organism because all the amino acids could be affected after that particular mutation. So why then can many mutations in a multicellular organism's genes be neutral, meaning that they don't do any harm to that organism? If you look at your genetic code, if you look at that nice little chart that we provided, some of you noticed even when we went through translation, is that if you look at that third base in a codon, is that there's many repeated sequences or many sequences that code for the same amino acid. So you can have some wiggle room in that third base of a codon, that if there is a different nucleotide, you could potentially still code for the same amino acid. And if that's the case, then you're not going to change that protein at all. There are some mutations that are not neutral that can be bad. And I'm going to have you watch this Nova Cracking Your Code Tay-Sachs. Either we've already watched that in class or you're going to look at that in the folder. All right, so we've talked about mutations can be neutral. Okay, they can have no effect on an organism. Mutations can be bad. All right, the example of Tay-Sachs, it's deadly and has dramatic effects on an individual. But mutations can also be beneficial. And mutations are really important and play an important role in evolution. If we think about our lovely insects, many of them have become resistant to different pesticides that we put on fields. So we need to continue to refine and develop new pesticides and herbicides because there are mutations, and then those become favored in a population because they make it so our pests aren't killed off by our pesticides. And then we've also talked about in our cells unit with bacteria that some bacteria have become resistant to chemicals, especially antibiotics. We have these superbugs. And there's a change in the genetic message, a change in that DNA that oftentimes becomes favorable, does not make them vulnerable to penicillin, amoxicillin, or our different antibiotics. That can then be passed on to the next generation, and that mutation is obviously favorable to those individuals. So to review what we've talked about, what I would like you to do is take this original DNA sequence, all right, and this is the normal, and I would like you to make some sort of mutation. Okay, you can select from a frame shift or a point mutation, so you can do a substitution, insertion, or a deletion. And what I want you to do is take this original sequence, and this is going to be your DNA, okay? So then over here, below my mutation part, you're going to do out what is the mutated strand, okay? How is it mutated? How is it different from this original strand? And then you're going to show me what is the mRNA strand, okay? Once again, you're still doing it out for this original or normal over here. And then you will also show the mutated strand, okay? And the mRNA strand of that individual. Finally, you are going to show me the amino acids. Okay, what amino acids are coded for this particular individual, and then what is it for the mutated individual. Finally, you're going to tell me what kind of mutation is this? All right, and what's the effect on this organism? Is it good? Is it bad? Is it neutral? All right, and you should be able to show me that with comparing your amino acid sequence from this individual from this individual. All right, so you can do that out. If you have questions, you should definitely talk with your teacher.